My name is Agneta Richter Dalfors. I'm a professor at the Karolinska Institutet, where I'm also director of the Swedish Medical Nanoscience Center. We have made a biomimetic neuron, and that means that we have made a device which mimics the function of a nerve cell, in that it can sense signals over here and very rapidly translate that signal to the far end in order to relay the signals in the neuronal system. In the future, with all the nanotechnology approaches, this can of course be miniaturized, so that you could think of this becoming an implantable device eventually.
Hello, I'm Ben Lewerton from the Swedish Medical Nanoscience Center. I'm a postdoc in organic bioelectronics with Agneta Richter Dalfors. So what we've built here, and I'm holding part of it in my hand there, it's basically an artificial neuron. And what we have here is the organic bioelectronic device that actually delivers neurotransmitters. And what we've done in this recent publication is we've actually connected this to a sensor part as well, so we can actually sense, transmit the signal and actually reproduce the effects that a real neuron would actually produce in our lab. So this material here, this is made of an organic bioelectronic polymer, and this has two different properties. Basically, it can transmit electrical signals, but it can also transmit ionic signals. And this is really interesting because this is exactly what a neuron can do. Hey. Okay, so now I'm actually going to explain the experimental setup which we have here. So in this petri dish, this pink liquid here, I'm going to pipette some chemicals that we can then detect, and I'll show you how that works. So now that I've pipetted some chemicals in there, we can actually sense that, and that electrical signal is then translated into an electronic signal, which then transfers into this machinery here. And these computers then interpret the electronic signal and decide if we should then produce another chemical signal at the other side of the neuron. So this would be the sensing side of the neuron. It then travels into the computers and comes to the microscope stage, where we then can release more chemicals to the cells. And so the computers can actually decide whether the cells will be stimulated or not. And we can detect that using the microscope. And we hope that this in the future will be actually used to treat neurological disorders. In our lab, we also see that there's lots of parallels with the use of organic bioelectronics to treat neurological disorders and to treat infections.